As long as we're talking about microorganisms, we should also talk about microscopy, or microscopy is what I say. I'm from the Midwest. We talk in a way. Anyway, microscopy, right. So what I want you to know about microscopy is its limitations and the major different types of microscopy. So what can we use them for and what can we not use them for? And what are they? So here's kind of the challenge we have. Biology happens on a scale that varies from a meter, like humans are around, whatever, a meter tall, 2 meters tall, essentially, all the way down to a nanometer, right? In physiology, you study proteins and lipids. Those are between 1 nanometer and 10 nanometers long. Bacterial cells are between 1 and 10 micrometers long. You definitely can't see any of these things. So how do we study them? It's hard even for microscopes to study them, but we have to. And if we're looking at this, I'm expecting you to know what an nm is and what a mu-m is and a cm and an mm. These are the important units. Meters, well, this should be review. Make sure you go through this because I will use these units a lot. And we'll do some calculations in the next lecture. Okay, so micrometer. This is about as long as a bacterial cell. They are roughly 1 micrometer long, if we broadly average them. And let's see. Okay. So what we have to think about with microscopes and with microscopes is their resolving power. That's the most important variable we have to think about. And resolving power is the ability to discriminate 2 different objects. That is, resolving power tells you if you have 2 objects a certain distance apart, can you say for sure that they're different objects, or do they blur together and look like one? So human eye has a resolution of around 100 micrometers. How many millimeters is that? How many millimeters is 100 micrometers? Why don't you just pause this video and come back and we'll make sure we're on the same page. 3, 2, 1, pause. While we're doing this, let's go back to this cool drawing and do a quick calculation. So I said 100 micrometers. How many millimeters is that? So I'm going to do a very explicit calculation. I'm going to multiply that by a fraction. I'm going to multiply that by the number of micrometers in a meter. Or actually, I'm going to multiply it by 1 meter divided by 10 to the 6th micrometers. 1 meter is 10 to the 6th micrometers. These are the same length. So I can multiply this fraction by this without changing anything. And this will allow me to cancel out the units of micrometers, and I'll be left with meters. Then I can multiply them by a conversion for millimeters. So I have 10 to the 3rd millimeters per meter. Right. And so that equals whatever it equals in millimeters, right? Because I can cancel micrometers, I can cancel meters, and then I'm just looking at powers of 10. I'm just counting zeros. 100 times 10 to the 3rd divided by 10 to the 6th, that is 10 to the 5th divided by 10 to the 6th. It's 10 to the minus 1. So 100 micrometers is 10 to the minus 1 millimeters. Now, if that doesn't mean anything to you, there's another way we could write it, which is 100 micrometers equals 0.1 millimeters. Okay? That's all I wanted you to be able to do. That's something you should definitely be able to do. It takes practice doing these calculations, but if someone gives you the conversion factors, you should be able to set these up. Though again, it takes practice. I don't expect you to be able to wake up and instantly think about how to do this like first thing in the morning, but you should be able to figure out how to do it. Okay, so the human eye has that resolution, 100 micrometers or 0.1 millimeters. That's kind of how small a gap we can see between objects. Lower resolution is better, right? Lower resolution, so a smaller number, is a smaller distance that we can see between objects. So we can put them closer together and still tell that they are two different things. So lower resolution is better. In a compound microscope, the type that you use in a laboratory to look at histology, and the type that I use in a microbiology lab, can generate resolution theoretically of 0.2 micrometers. So that's about the best you can ever expect to get with a compound microscope. Compound microscopes come in a lot of different forms, and these are what you have to know. Bright field, dark field, phase contrast, and fluorescence. These are all different ways of using compound microscopes. And then once we've gone through those, we will look at electron microscopes. So compound microscopes, you should recognize them. They have ocular lenses you look through, objective lenses that are close to your sample, a stage you put the sample on, focusing knobs, stage controlling knobs, stage clips, what else? Condensers, the diaphragm that controls the amount of light that gets through. Okay, all compound microscopes have these broad parts in common. They have a light source of some kind. They all have this in common. They're going to differ in some of these details. They all have the same way of calculating magnification. So the magnification, this is the other thing you have to think about. You have to think about resolution and magnification. And you've already thought about magnification when you've done histology. And again, the total magnification is the ocular times the objective. Most oculars are 10x magnified, and your common objectives are from 4 to 100x. A 100x objective requires immersion oil. And that becomes important in microbiology because you have to use this all the time because it's the only one that can show you bacterial cells because they are tiny. So these are three different objective lenses showing kind of what controls, what controls how they work. So the idea is light is coming up through the sample, and some light is going this way, some is going that way, some is going that way. Any light in this cone goes into the microscope. So let's remember bacteria tend to be half a micrometer wide and 1 micrometer long, roughly. This red oval is what's in focus for these different lenses. And let's get into it. So our lowest power objective, 4x, it gives us a resolution of around 2.7 micrometers. It also gives us a good depth of field. It's easy to focus this objective. And if we're trying to look for a bacterial cell that's 1 millimeter long, this magnification, 40x total, 4 times the ocular 10, 40x magnification, that 1 micrometer long cell will look like it's 40 micrometers long. we have 100 micrometer resolution, so we can't really see a bacterial cell using this objective. The 10x gives us better resolution and more magnification. A 1 micrometer cell might look just about barely visible to us. But if there were two of them a micrometer apart, they would be blurred together. That's why we have to use the 100x objective. It has a really, really narrow focus distance, so you can easily be out of focus. You can actually focus on the wrong side of the slide by accident when you're using this this objective. Notice, if I go back, how sharp this angle is compared to this angle. The 100x objective has to be very close to the sample, and so it looks at light at this very wide angle. And it can give us overall 1,000 times magnification. So a 1 micrometer cell looks like it's 1 millimeter long to us, and we can see that. So in a perfect world, that 100x objective could resolve objects 0.29 micrometers apart. You have to push a microscope to get that 0.2 resolution. And typically, we can think of it as closer to this. With that extreme angle, so again, that extreme angle, typically not enough light actually gets in, and so you have to use oil, or else it's too dark to see your sample. And the problem is that the interface between the glass and the air bends the light. And so if we look at this slide, look on the right, this is showing a light path in air, and a typical light path going through the condenser hits your sample and then bends away from the microscope. You don't see it. So a large fraction of the light going through our sample we just miss if we're using a 100x objective in air. If instead we use immersion oil, it mimics glass, and so the light does not bend. So that's this here. The light comes out this way through the sample. It doesn't bend. It goes into the microscope. So if you use oil, you get more light. It's brighter, and you can actually see your sample. And so I just showed you that because that really is your nemesis if you're in a physical microbiology lab. The hardest thing you will do, literally the most challenging thing in a microbiology class, is learning to focus a 100x objective lens on a thin preparation of bacterial cells. It is 100 times harder than looking at a histology slide. So there's that. Okay, so what are the types of microscopy? Well, the one you're familiar with, if you've done histology, is bright field microscopy. This is where we observe stained specimens or very pigmented organisms. Bright field microscopy gets us a lot of light, but it doesn't give any contrast to our samples. So any cells you look at have to have their own contrast. They have to be dark and colorful. This is the least expensive kind of microscopy, and it has this resolution around 0.2 of a micrometer. If you get a classroom microscope and it's only going to be used for histology, you can get a bright field microscope for less than $1,000. Dark field microscopy is very different. It uses tricks to increase the contrast. So for dark field microscopy, the condenser has an opaque disc that blocks the light completely. So the only light you see is light that is bent by your sample. So you see a dark background with light specimens. So this is really useful if you're looking at living organisms that are big, algae, protists, and stuff. You can do this with, you can get a 100x dark field objective, but it's really expensive. I've never actually seen one in a laboratory. I really wish that this would be what I want for Christmas, would be a dark field 100x objective. But they cost as much as a whole microscope. I think they'd be $2,000 just for that one lens. It annoys me a little bit that in Labster, they will tell you that dark field microscopes are the most useful when they're not. In microbiology, we just don't really use them unless we're studying some big eukaryotic cell, which we'll talk about later. What we do use all the time is phase contrast microscopy. Here, the condenser has a ring-shaped opening, and I don't even want to get into how it works, but you can get constructive interference or destructive interference. And what you get is dark cells with a light background. So you can look at living bacterial cells using this technique. And the cells will appear dark and the background will appear relatively light. And the idea is that cells are full of essentially salt water, and as light goes through, it goes a little slower through the salt water and it gets out of phase with the background light. Phase contrast microscopy can go up to 1000x magnification. So that is a 100x phase contrast objective with a 10x ocular, and this is common. A lot of microscopes in teaching labs have them. A lot of times the scopes you use for histology will have been purchased to be both for histology and microbiology, so you'll actually see that you have phase contrast objective lenses. They are more expensive than bright field, but they're less expensive than dark field. So these are routine. So a microscope, a bright field microscope might cost $1,000, and the 100x phase contrast objective would also cost $1,000. And so that phase contrast objective would sometimes be the biggest expense in a microbiology lab. Dark field would be about twice as expensive. So this is what it looks like. A really good microscope. This is from my wife's lab. It's a really, really, really nice microscope doing phase contrast microscopy. It looks like this. Little tiny bacterial cells. You can't see much detail. These are 1 or 2 micrometers long, and the resolution is 1/5 of a micrometer. So there just isn't much detail you can see, but the cells appear. If this had been bright field microscopy, the cells would be very faint kind of ghosts. You wouldn't really see them. So here is a summary of the 3 different types of microscopy that I've mentioned: bright field, dark field, and phase contrast. And the authors of a different textbook, well, your textbook authors have given us these diagrams that show how the different scopes work. And then they show us how a giant pigmented protist would look under these different things. So I went to a different textbook that shows us how moderately sized yeast cells, unpigmented yeast cells, would look under bright field, dark field, and phase contrast. So the simplest kind, bright field, doesn't add any contrast, and the yeast cells are hard to see. You can't see much detail because they are just bags of salt water, and salt water is invisible, right? Dark field microscopy gives us this dark background, and the cells bend the light, so you see them glowing. Phase contrast microscopy is different. These are somewhat dark against a lighter background, and realistically, it would be better if we had examples of what bacteria look like. with these, but nobody has a 100x dark field objective for looking at bacteria. So this is what they look like in the lab because we use phase contrast in microbiology labs. Okay, those are techniques for seeing the size and shape of microorganisms. That's about all you can do with any of these. You can't learn much from the size and shape, so you just say, okay, they're round, they're a certain size, and that's about all you can get from any of these. Dark field and phase contrast, you can look at living cells. So if they're swimming around, you can see them swimming. Whereas bright field, typically you would look at stained samples. So this picture is not what you want to see. You want to see much better contrast. So bright field you'd use more for like histology slides. So if we want to learn more about microorganisms, we're going to use fluorescence microscopy. And the idea here is that we can detect very specific things on the surface of a cell by getting antibodies that attach to that particular thing on the surface of the cell. So the antibodies are only going to attach where that specific thing is. And so we can use it to figure out where it is. And we can attach the antibodies to a fluorescent dye. And so that's expensive. You have to buy very high-tech antibody dye molecules, and you have to get very specific optics with very specific lenses. And this is easily $100,000 worth of microscope. But this is by far the most useful kind of microscopy in research. Because you can use this to figure out what is on the surface of cells. And you can get the same magnifications that you can in any other compound light microscopy, because this is just light. The difference is, in fluorescence, you get light of a certain color is absorbed by the dye, and then it's emitted in a different color. And that allows us to see very specific things against a very dark background, so it appears to glow. Like this green light, I believe the light that is being shined on these cells is blue, and they filter it so that you can't see blue light. So that makes the background totally dark, and you can only see green light from the fluorochrome, and it only appears wherever this antibody with the fluorochrome attached, wherever it has attached to the cells. So in this case, it's lighting up the entire cells, but you could just as easily have gotten an antibody that finds one little rare thing on the outside of the cell, and it would only show up here, and that is the type of thing you might do in research. Okay, so fluorescence microscopy is a type of light microscopy. Again, you're using visible light. It's a type of compound microscopy. You have an ocular lens, an objective lens, a condenser, different things like that. Bright field, phase contrast, and dark field are all different types of compound microscopy using light. So that's all light microscopy. Electron microscopy is fundamentally different because it uses electrons instead of light. Electrons have a much shorter wavelength, and that gives us much better resolution because the wavelength is what determines the best possible resolution. So with that much better resolution, we can get better magnification and really see little tiny details. Unfortunately, electron microscopy doesn't let us look at living cells. We can only look at dead, fixed cells. And unfortunately, it's extremely expensive and complex. And there are two types of electron microscopy you need to know. One is transmission electron microscopy, and this is the most high-resolution type of microscopy we use in biology. you can get a resolution down to a nanometer. But you have to embed the sample in a block of metal, and you have to use special electron deflecting stains. So you have to get an antibody and attach a gold particle to it or something like that. You can get really high magnification, really great resolution, and this is how you can study the structures inside a cell. Again, you can use an antibody to attach to something inside a cell, and use transmission electron microscopy to see a cross-section of the cell. The other type of electron microscopy we use is scanning electron microscopy, and it gives us a 3D picture of the surface of a cell. To do this, instead of embedding the sample, we coat it with metal. So we put it in a machine that coats it with platinum or gold. I've used both of those. And shoot electrons at it and then detect sort of what happens. This has great resolution, much better than you get from light, but not quite as good as what you get from transmission electron microscopy. And these are two false colored pictures of a large eukaryotic organism, like a paramecium or something. This is not a bacterium. This is much bigger than bacteria. Scanning electron microscopy lets you see surface features like the little hairs on the outside of this thing. Transmission electron microscopy lets you look at a cross-section and you can see internal structures. And they've gone in and colored the structures either yellow or white or red. And that's it for microscopy. That's all you need to know about microscopy. Be able to tell me the differences between the 4 types of light microscopy, the 2 types of electron microscopy, and be able to tell me what would happen in light microscopy if you used a 100x objective without oil. And that's enough for this one long video.